Okay, the floor is yours.
Okay. So thank you for coming. Yeah, as Jörn noted, the primary article from Jumper et al., the paper that would later lead to a Nobel Prize, is a little challenging to read. But that article has a nice introduction, the first page or two, and then I think he might have included the commentary also from Jumper and Hassabis, and in that article you'll have more detail about the general approach taken in AlphaFold. So I won't be talking only about AI. I will use some examples from my team's research to explain why these tools are so important to us, whether you're working in plant molecular biology or medical sciences, or frankly any domain in the life sciences is touched by these tools. So I'm over at the IBMP, and our group focuses on a family of enzymes known as RNA polymerases, and I'll tell you more about that in a minute. So these tools today from artificial intelligence sometimes get associated with various memes in society, such as evil robots, or very artistic depictions of a brain with lots of little lines connecting together digitally, or some scientists in a lab doing something mischievous. And there's some truth in each of these images, but the actual reality of artificial intelligence that's used in most applications in biology is more closely related to large language models. And you're familiar with these. You've been using them to study or write essays and many other things. They're very carefully designed to be interactive. So these chatbots such as GPT, Mistral in France, or DeepSeek that was recently produced by a group in China, they're incredibly powerful, and what they are is all the documents on the World Wide Web. It's almost uncountable how many documents there are in the internet, but they're being sampled using different databases. They're accessible. For example, the Common Crawl is an open source database of 2.6 billion pages from the internet. You can download it right now. You don't have to pay anyone for this. There's Wikipedia, of course, which is free and open. Digital books. Of course, these companies have been training them also using proprietary material, which is an ethical question. Scientific articles, code repositories, and so on. And then they break the data, the text, down into what are called tokens. After filtering and deduplicating them. The tokens look something like these colored highlights. You'll notice that they don't fall neatly on word boundaries. So you may have half a word in a token, you might have punctuation in a token. So the large language models are trained on these billions of word and partial word tokens. And then the goal after training is for the LLM to predict the probable next token. It's really that simple. In reality, in the implementation and the engineering, it's more complex. But the idea is, after a series of tokens, say, you come across efficiently. And the question is, what comes after efficiently in the response that a user would like to have? So in this case, it's efficiently representing common words. So the tokens in red have a next token in blue. And of course, this is on a very large scale. The context window in which the large language models work can be thousands, millions of tokens. So the next point is that they're not handling them as words. So in the computer code and the algorithms, they're not able to handle human readable text. What they have is the tokens, the dog sat on the couch from this sentence here, broken into indices, so they would be numbers. And then those numbers are encoded in vectors, usually where there'd be 0, 0, 1, 0, 0, and so forth. So these vectors can be combined into matrices. And that's, these matrices, that's what's being acted on when the large language models are being trained. So this is the, if you will, the input to the process of creating an AI or an artificial intelligence tool. So just like there was a lot of complex explanations in the Jumper et al. Paper published in Nature, I don't expect you to understand all the details here, but the basic point is Matrices are best handled by a type of computer chip called a graphics processing unit, a GPU. So these GPUs have thousands of individual cores in them that can process data. They're suited for parallel workloads, and they're excellent for matrices. So if you have matrix calculations, you want a GPU. Finally, they are dependent on the normal central processing unit. So every one of your computers has a central processing unit with 8 or more cores in it these days. The idea is you have some data. So these would be the indices, a matrix of the data from the large language model or protein amino acid information. And you know from labeled data, data that you understand already, that you would expect a 1 as an output when you have 7, 8, and 10 here. So the idea is you have layers of neurons that are connected, and these connections can be weighted, and you will eventually, after doing a calculation across this network, end up with some value, some prediction. So you said that when you enter 7, 8, and 10, you expect to see 1. And the problem is when you enter 7, 8, and 10 and you did this complex calculation, you ended with 0.521. So you have a prediction that's not very accurate. So what happens is you can adjust in the algorithm, in this training of your large language model, you can adjust the weights. Those are the things that connect these different nodes together. And so there are weights here in W, and then there's biases in B. And so these different numbers can be adjusted, fine-tuned, or tuned by the training. As this happens, as the weights and the biases are corrected, you can get closer to the value you expect. We expected 7, 8, and 10 to give us 1. And by training this model, we can end up with 0.998. So that's much, much closer to what we expected. And this is done over many cycles. So many epochs of training or cycles of training, we get closer and closer to the right value. And this is how these artificial neural networks work. Now, there is some differential calculus involved. We do a forward pass to see what value we get after these calculations are made, and then we have to go backwards and see how to adjust. So there's some linear algebra and more complex differential math calculus involved. But the main point is we can do this very reproducibly on all kinds of data, whether it be the tokens representing words from your text online or tokens representing amino acids in a protein. So the main point here is that the weights that are connecting these different values together, and then something called a bias that is used during another part of the calculation, they are called parameters. So the weights, the number of weights and the number of parameters, they have something to do with the complexity of the model. You can count up the weights and the biases. And if you look at how complex the large language models are that you guys are using to better understand the world by asking questions and getting responses, we can see how they have evolved. We are adding parameters to these AI models and improving their performance. So the GPT-1, which was released in 2018, only had about 117 million parameters. So the little diagram I showed you had 10 parameters. Now we have 117 million. And this was also a new architecture, a new way of training models. It was called a generative pre-trained transformer. So something about the way that those neurons were arranged in this artificial neural network was special. But in the end, the idea is if you keep updating these models and adding more parameters, 1.5 billion, 175 billion, or greater than 1 trillion, which is what GPT-4 used, we know that we can get closer and closer to results that seem human in terms of text, and that we can be better at showing some aspects of analysis or cognition. It's not thinking and it's not general artificial intelligence, but when you talk to these models, when you chat with them, you get a feeling that they're actually responding to your request in a human-like way. So there are additional aspects. The GPT or generative pre-trained transformer architecture has gotten more complex to add a mixture of models. So these different models are called experts, and these experts are being trained in parallel, but the idea is the same. You add tokens. Those tokens can have a certain value that is high or low probability after each other, and then over time you can string together sentences that have meaning. So the problem, of course, is the cost. So this is not something that you can do in your own apartment or even with your own research group. It is an industrial scale kind of AI research. So GPT-3 probably cost over 4.6 million euros, and it also cost 114 megawatt hours. GPT-4 was even more, so well over 100 million euros cost to train. 50 gigawatt hours, which would really power a medium-sized European city for months. So this is a lot of energy, a lot of investment. It also brings with it carbon costs. And there's a question about the ethics of how many of these large language models should be trained because of the energy, the money. There's always an economic and an environmental cost. And of course, these values here in terms of 50 gigawatt hours of power for GPT-4, that's just the training when you're using the model, when you're querying the model, when it's being, it's serving you data, then this also costs energy. So I would like to connect this use of large language models to what we need in molecular biology. And to define that a little better, I'm going to talk about molecular biology and how it arose. So the founding discovery in molecular biology, some would say, is the DNA double helix, which you can see here to the left. This is the original model. It was built out of metal pieces, handcrafted. It was very artisanal. You can see here an adenosine that was crafted, of course, crafted carefully. This is not just a tinker toy model for children. These geometries, these distances, they were measured precisely. So this model could be realistic and give the researchers some knowledge about what a double helix would look like. Today we know how the T and A and G and C work together because of this discovery, which was the fruit of the labor of Rosalind Franklin, Francis Crick, James Watson, and others. There's a whole historical aspect I won't go into, but one thing I'd like to point out about this early discovery in molecular biology is how young everyone was. Rosalind was around 32 years old, and Watson was only 24, and Crick was 36. In other words, Crick was the old one. So these are people that are just a few years older than you or your age. They are people that are interested in molecular biology. They're striving to understand the deeper secrets that were hidden in the hereditary molecule. And I think we can take inspiration from the youth of these researchers that made such incredible breakthroughs. Because the double helix was important for the mechanism of inheritance, the genetic code, functions of biomolecules, causes of disease, and evolutionary history, and many more things. So it's the foundation of many research programs just a priori. But of course, there's a whole context today. We understand better that, that in molecular biology you have DNA wrapped around nucleosomes, leading to chromatin. And then if you unwrap that DNA, you can look at the DNA sequence, but of course this is happening in a larger context of the cell. The DNA is in the nucleus. There are different levels of chromatin compaction. You have the nuclear pore complex here, allowing molecules to come in and out of the nucleus. And so that's where you can see all the exciting topics, and even in modern molecular biology, is how all these features work together. You have gene transcription on the DNA leading to RNA. You have the ability to regulate these genes through promoters and enhancers. This molecule, RNA polymerase II, or this complex RNA polymerase II, is one that we study in my lab. And then with the messenger RNA that is synthesized off of these DNA genes, you can exit the nucleus, and the export of the messenger RNA leads to translation, protein synthesis, and you kind of have the whole central dogma of molecular biology here in one illustration, leading to proteins that fold and have functions in the cell. So how do we use tools of modern AI or machine learning to address questions in this context of molecular biology? Well, one way is we can look at the DNA here and the RNA there using Oxford Nanopore sequencing. So Oxford Nanopore sequencing is a small device. It would fit in the palm of your hand. It connects to a computer, laptop, for example. And then in this flow cell, the DNA here, double helix, can pass through a motor protein and then through a small opening in this biological pore that is just wide enough, 1 nanometer in diameter, just wide enough for DNA to pass. So one strand of the DNA passes, but because there is a potential across the membrane that this pore is embedded in, we get changes in the current as these different nucleotides pass. And we can read from the changes in the current, this electric signal tells us something about whether we have an A, a C, a G, or a T. So we can sequence DNA this way. But in addition, the AI tools are allowing us in the community to look at nuclear pore complexes in much more detail, using even fluorescence microscopy. So this is some work done out at the IGBMC, some really beautiful studies where you can look at this, the ring of this nuclear pore complex, and using super resolution techniques powered by AI, resolve features that normally wouldn't be visible under a microscope. And then finally, the paper that I supplied to Jeroen today about AlphaFold is you take the primary amino acid sequence of a protein from the N terminus to the C terminus, and you can predict with high accuracy the protein structure. And this is a 50-year-old problem, something that had really been elusive in computational biology for all these years. So how does it work? Well, it's a bit similar to the question of how you break up text from the internet into tokens and then process it. You have to have a lot of data. So the data that they used at DeepMind to solve this problem was over 250 million sequences from UniProt and then some other databases you may not have heard of, such as Mignify, BFD, which is the big fantastic database. I think originally it was a big effing database, but for purposes of appearances, they changed the name to the big fantastic database, and it covers over 2 billion protein sequences. So that adds to this set of non-redundant sequences that they're going to use. MSSEQ2, PFAM. So these are sets of aligned sequences in some cases that have been already investigated through protein alignment, and they're kept in databases that are standard to this sort of protein prediction approach. But of course, those are linear, so primary sequences of proteins. They also have used greater than 100,000 experimentally determined 3D structures from X-ray crystallography or cryo-electron microscopy. So that's been stored in the PDB, the Protein Data Bank. And they're combining these in a special artificial intelligence architecture that's based on the transformer architecture used for the large language models like GPT-3 and 4. But it's been modified to allow for insights from evolution and three-dimensional structure of proteins. So they called it an evoformer to call attention to the fact that it's like the transformer models used in large language models, but it has some adjustments and it's been specialized for this task. And a key point that I'll just mention without any technical detail is self-attention. This is a big part of the large language models that you interact with in a chatbot, but also for how AlphaFold works. Self-attention means that as you go through a series of words in a paragraph or a series of amino acids in a protein, you try to understand how those words relate to one another or how those amino acids in the protein relate to one another. This can be in 3 dimensions or through their evolutionary history. So the idea isn't that you just predict one token after another and then forget what happened before. That wouldn't work. The models that work for chatbots and that also work for AlphaFold remember some of the context of the text that came before or the amino acids that came before in the sequence. And so there are aspects of the model, parameters, which were being entrained to pay attention to this fact. So this attention to the pairwise relationships between amino acids is what makes AlphaFold work. And then you need these GPUs, graphics processor units. Now for AlphaFold, it happens to be NVIDIA. They are a dominant player in the marketplace. Luckily today, the AMD GPUs and the GPUs that are in your Macintosh, your Mac laptops will soon be accessible for these sorts of tasks, but originally it was NVIDIA. So I want to mention the parameter count. To do this training of AlphaFold, they only needed 93 million parameters, which is quite a lot fewer than are used for the large language models. And then what you see here is the amazing results. I'm just taking one example of a particular PDB file. So this would be determined experimentally in blue. And you can see how AlphaFold has predicted in green with nearly no deviation, a very high quality three-dimensional structure of this protein. And then on the right, this is the sort of thing you need to do work with models like this, to have confidence in them, is some sort of metric of your confidence. How much do I believe, or does the model, does AlphaFold 2 believe in this prediction? So this is called the predicted local distance difference test. And what you're seeing here is a scale of 0, which would be in red. You would have no confidence in that region. It could be intrinsically disordered or not structured. As you approach the 70, the color turns green and then blue. And as you pass 70, 90, 100, you're in a range where the prediction can talk about domain structure and even individual amino acid orientations with high confidence. So this has been very powerful in the larger community of molecular biologists and biochemists. And I'll just cite one example in a minute, but just for comparison, as you recall, GPT-1, 2, 3, 4 moved from 117 million to 1 trillion parameters. In AlphaFold 2 and 3, they're more in the tens of millions. And this has to do with the fact this is a more specialized architecture, so it's more focused on these evolutionary relationships and amino acid pairwise distances. So the AVO-Former allows you to work with somewhat fewer parameters than a large language model and get good results. And in addition, that also means it's not as expensive or as energy intensive to train. Still something that you probably wouldn't do in a small research laboratory. It really requires some company support to do this work. So this is a really beautiful example that the authors themselves of AlphaFold cite in some of their interviews. And it's basically a human nuclear pore complex, the one I mentioned before, that serves to the translocation of different kinds of molecules in and out of the nucleus of a cell. And there is some primary cryo electron microscopy data that is being used here, but there were so many different protein subunits in this ring, they didn't have enough resolution with the primary experimental data alone to know how to fit them. So they used AlphaFold 3 to predict individually many of these subunits and the different bright colors. And then they could then, with high confidence, once they had the predictions from AlphaFold, they could dock them into the overall structure to produce this very, very beautiful depiction of the cage and the ring around the NPC. Last time I spoke was last year. We didn't know that this had happened yet, but I had a kind of a feeling, I think a lot of us in the community had a feeling that this would lead to a Nobel Prize fairly quickly, as it did. So first of all, from the AlphaFold team, you have Demis Hassabis and John Jumper, who were the first and last authors of that important paper in 2021. But in addition, and I think this is equally important, they shared the prize with David Baker, who had been working on methods to understand how protein structure could be predicted some decades before. So this is a large community of structural biologists and computational scientists who had already been working deeply on this problem for decades. So it's not just the AI companies that jumped in. There are researchers at universities that were participating in these huge discoveries. And in the case of David Baker, it's interesting, his Rosetta tools were originally designed to say, if I want to know the sequence of amino acids I would need for a structure I'm interested in, what would that sequence be? And the AlphaFold people were asking, if I have a sequence of amino acids, how would I know the structure? How would I predict the structure? So these are, in fact, synergistic approaches. One is more from an engineering point of view, in the case of David Baker, and the other is from the fundamental research point of view of exactly what do these primary sequences look like when they're folded. And I do recommend, if you have time, to look at this YouTube video that was sponsored by Quantum Magazine. All right, so we know that AI tools are important in genomics and molecular genetics. One case I'll talk about in a minute is epigenetics. I mentioned the structural biology uses in drug development, 3D structure, protein DNA, RNA complexes, and also simulations of molecular dynamics. And a field that I won't talk about, but it's equally important, is advanced light microscopy, so cell imaging, super resolution, image processing, segmentation, and so forth. These are not an exhaustive list. But I want to mention, for the work we do in my team, how we've made use of 3D structure prediction and tools for epigenetics. So we work on plants, and in those plants, in the 1940s, Barbara McClintock and Mays noticed there were some pieces of DNA that are mobile, and they're independently mobile from the chromosome itself. That is, they can cut themselves out and then reinsert them in new locations. They're known as, today they're known as transposons. And they make up a really big part of the human genome, 45%. In Mays, it's even larger, 85%. And in some of the model species we work with, Arabidopsis, which is a eudicot, and Brachypodium, which is a monocot, they make up a 20 to 30% of the genome. So they're a very major constituent of genomes. And of course, if they insert in the wrong place, in the wrong gene, they can cause some detrimental effects. That kind of potential deleterious mutation is the sort of thing that organisms have evolved to avoid. So there are cellular machineries that can stop this process of mutation through transposon mobilization. One very important group are the epigenetic modifications, so repressive chromatin modifications that occur on the nucleosomes, or cytosine methylation, which is just a single methyl group added to the 5th position on a cytosine ring. So 5-methylcytosine and repressive chromatin modifications, they can lead to a compaction of chromatin, lead to less accessible genes that can either be silenced or at least repressed for a period of time. And this is particularly true of these transposons. So silent transposons tend to have DNA methylation and repressive chromatin modifications. And the issue there is how you maintain this information. So one family of enzymes that do it are called maintenance methyltransferases. So if you have a methyl group here in this cytosine on this top strand, and then another methyl group on this cytosine on the bottom strand, this is known as CG methylation. And over the course of DNA replication, there are new daughter strands that form here in green. They're complementary to the parent strands. This is this semi-conservative replication of DNA. And you'll notice now that we're missing the DNA methylation. But this is often a way of conserving genome integrity and sometimes regulating genes. So then you need to maintain those methyl marks. So an enzyme called MET1 comes in and it marks the daughter strands with cytosine methylation as well. So you end up with these CG dinucleotides marked again. So this maintenance is a form of epigenetic information. It's inherited after cell division, either through mitosis or even in plants in meiosis. You can inherit these through generations of plants, seed to seed. And I think there's a little bit of issue with the images here, but I'll just mention what's going on. So in the wild type case, we can maintain this generation to generation. But in the case of a knockout for the MET1 enzyme, the levels of methylation drop off over time, and then you end up with a lethality. So the plants are no longer able to survive. So you have this need for methylation on the DNA, but the question remains, how do we know what parts of the genome should be methylated? You know, what guides the DNA methylation? What silences the transposons? And in my lab, we're interested in exactly that topic. An enzyme that can recognize potential targets, transposons in the genome, and by doing so, it can repress them. And this enzyme evolved in plants about 450 million years ago. So normally in all single-celled organisms, such as bacteria and archaea, you have one RNA polymerase. And then in eukaryotes like us, you have pol 1, 2, and 3, so you have 3. Plants happen to have a more diverse set of enzymes. They have pol 1, 2, 3, 4, and 5. This is unique to plants, and it's a really amazing discovery of about 20 years ago, is that terrestrial plants have these extra RNA polymerases. And they look a bit like RNA polymerase 2. So remember, RNA polymerase 2 is what's transcribing DNA to make messenger RNAs. But we know from a lot of work, including work I've done during my postdoctoral studies, that POL4 and 5 are not like POL2 in that sense. They do not synthesize messenger RNAs. So here's POL2 on the left, synthesizing pre-messenger RNA, the splicing, capping, and so forth. You have the messenger RNAs. POL4 transcribes DNA, but then immediately after that, an RNA-dependent RNA polymerase, which is it's an enzyme that can act on a template RNA to make another strand of RNA. So you make double-stranded RNA. And this double-stranded RNA is then cut into little pieces called small interfering RNAs. So this is a hallmark for a kind of RNA interference, silencing of genes. It occurs in the nucleus of plants. And then the other enzyme, which I won't mention much in my talk today, POL5, is able to make long non-coding RNAs that, again, are not acting in terms of coding for proteins, but they're involved in gene silencing. So together, the small interfering RNAs here on the left, made by POL4, and the nascent non-coding RNAs made by POL5 on the right, they're involved in this system of transposon silencing. And they're connected together. That is that when small RNAs are made from one enzyme, they can pair, using an Argonaute protein shown here, to the transcripts made by POL5. And when this pairing occurs, this is a signal that this would be a region of the genome that you should keep silent. It could be a transposon. And we've been studying this in various models. I will briefly mention one, which is Brachydipodium. It's a cousin of wheat and maize. It's important as a grass species throughout the Mediterranean. And one of our collaborators, Anne Roland, at the University of Zurich, she's looked at these populations of Brachydipodium all around the Mediterranean basin. So if we knock out this enzyme called POL4 using different kinds of genetic mutations, we see these small interfering RNAs that we detect here by northern blot are gone. And when this happens, then of course we expect retrotransposons to become activated. And this is what happens. So we can do RNA sequencing and get these transcripts here forming that are coming from retrotransposons. And you can even see the retrotransposition events occurring in DNA circles that you won't have in the wild type, but you will see in the POL4 mutant. So these are extra chromosomal pieces of DNA that are transposons, retrotransposons, that are able to reinsert in the genome. So we discover here the active hoplite retrotransposons in Brachydipodium, but also noted that these elements are actually activated after fungal disease infection. So there's a link between stress, such as pathogen infection, and the activation of these elements. But another thing about these plants, these POL4 mutant plants that interested us were that they are smaller. We can see that their growth is slowed. After a few days, we start to see a plateauing off of the mutant for POL4, and the wild type outcross and inbred lines, they're just fine. So they're growing in a normal way. This is highly reproducible, and it suggests there's something more than just controlling transposons going on. We think that POL4 is somehow controlling growth. And so this is where we started to look at our options in terms of detecting the DNA methylation I mentioned earlier. How do we detect it? The original methods from 2008 and afterward were called whole genome bisulfite sequencing. And basically, you treat your DNA with a chemical, bisulfite, and that changes one of the bases, cytosine, if it's unmethylated. If cytosine is unmethylated, you end up with a uracil. If it's methylated, then it stays to be read like a C. And we can then do short read sequencing using a technology called Illumina and see all these short reads and detect the DNA methylation. On the other hand, there's a very short reads, and it's hard to put them back on the genome of Arabidopsis or Brachydipodium in this case. So to better do that, we're using the nanopore sequencing technology I mentioned, in which the DNA passes through this small pore. You have changes in current. These are read out onto a computer, and the computer can detect not only the A's, the C's, the T's, and the G's. It can differentiate this fifth base, 5-methylcytosine. So you can differentiate between a methylcytosine and a cytosine. You don't have to make any chemical treatment or fragment the DNA. You can work with long reads that are tens of thousands of bases long and then more easily assemble the genome. And again, this technology depends on NVIDIA GPUs, on deep learning, and the technology I mentioned before for protein structural prediction. So these are a common theme. You come back to having a lot of data that are being trained on a model, and that model allowing you to see new information, in this case, a CH3, a methyl group on the 5th position of a cytosine. So we use this technology and we're able to detect, lo and behold, a transposon inserted in front of a particular transcription factor gene here, this BAH-LH gene. We see it in the Illumina data, we also see it in the nanopore data. And in our POL4 mutant, this methylation is gone because the idea is that POL4 makes small RNAs. The small RNAs target a region of the genome and then it becomes methylated. So we're using, in the case of the small nanopore sequencer, a completely different technology to reproduce and confirm the data of where this methylation is present in the genome. And so, interesting enough, when we look at that transcription factor, BAH-LH transcription factor, we see it is highly overexpressed by RT-qPCR. We see a lot of transcription of that gene in the POL4 mutants. It seems like it's deregulated. So this transcription factor is our best candidate to better understand why growth is inhibited or changed in the POL4 mutant, but occurring normally in the wild type. So to summarize this biology part, we validated an allelic series of mutants that POL4 silences transposon proximal genes through methylation. And POL4 controls the accumulation of this transcription factor, but we're still looking into it. Some studies we've done that I didn't tell you about today shows that this transcription factor attenuates along the longitudinal axis of the leaf. So it seems to be also expressed differentially in the leaf. That might explain why if we allow that transcription factor to be overexpressed, we might affect growth. We have natural epiallelic variation occurring at these genes. So our partner in Zurich, who studies populations, has sequenced many populations of Brachipodium, this plant here, and we're able to see that DNA methylation varying across them in the Mediterranean. Finally, clearly Oxford nanopore sequencing is highly suitable to look at these epialleles. So what happens is that the POL4 effect is to repress a nearby gene nearby which transposon has inserted, and this allows for some control of growth. So I mentioned the vegetative growth phenotype in Brachipodium. I'd like to also just briefly give a shout out to other work we're doing in the lab, which is that if you mutate this enzyme POL4, you end up with enhanced symptoms during viral infection. So when we infect wild-type Arabidopsis plants, we see some recovery of healthy leaves, and this does not occur in the mutants for POL4. So it appears that in addition to growth control and transposon silencing, POL4 has an important role in dealing with disease tolerance. So, but the idea would be this occurs to the targeting of POL4 to particular regions in genes. That would be the way that it could control growth and affect pathways other than transposon silencing. So to better understand that, we've been trying to figure out what makes POL4 go to particular sites in the genome. So POL2, we know that it is recruited through general transcription factors. In your general molecular biology classes, you probably heard of TATA binding protein or TF2D. These are complexes of proteins that sit on a promoter. When they've been recruited there, then POL2 would be also recruited and you have transcription. Now, we don't have that with POL4. The POL4 enzyme itself does not interact with those general transcription factors, but it does interact with these specialized chromatin remodelers that go by the name CLSY, CLASI. And until recently, we didn't know why or how those could interact with POL4, because POL2 and POL4, they're highly related. It would be interesting to know how POL4 could evolve to interact with them. So we had a student in the lab some years back who discovered that if you mutate a particular amino acid in POL4, in its largest subunit, you get a change in a conserved amino acid sequence, this motif. And that change leads to a loss of DNA methylation in this mutant and a lack of transposon silencing. So in this Arabidopsis plant, you have POL4 being disrupted just by a single amino acid disruption, leading to a loss of the DNA methylation and the transposon silencing. So we wanted to know why this is occurring. It was a bit mysterious because when we looked at a structure from a group in Shanghai of RNA polymerase IV, you can see in salmon color the subunit we're interested in. That's the core catalytic part. You can see below the RNA-dependent RNA polymerase that is accepting RNA as a template and then synthesizing double-stranded RNA. So you synthesize a single-stranded RNA by POL4 in the top, and then you directly add this, you allow this product to be channeled into RDR2 to make double-stranded RNA. But we couldn't find the region that we had mutated in that previous study. So this was the work of a student of ours, Louisa Felgin. Louisa Felgin is just completing her PhD this year, and she's done a lot of work in collaboration with Laura Martins at the Salk Institute and structural biologist Florent Waltz and the Biocentrum in Basel to better understand this work in collaboration with other members of our team. And what we were able to see was that there was a missing region in the cryo-EM structure that had not been resolved, and it corresponded to a particular region that we found mutated and highly conserved in POL4 but missing in POL2. And using this AlphaFold prediction approach, we could model this region here in 3 dimensions, and it revealed what's called the clamp head of the RNA polymerase. So the RNA polymerase has a domain that can fold over DNA on the template, shown in blue. And interesting, this AlphaFold structure also revealed why the particular amino acid we were studying in the previous paper is so crucial, is that if you mutate this cysteine in this high confidence region, so I remember blue means that you have a high confidence from AlphaFold, you can note that that cysteine is coordinating zinc. So this structure would be lost. This cluster of cysteines that are coordinating zinc would be lost in our mutation. And finally, we were also able to find evidence in a low-pass filter of the electron densities of the original paper, some evidence for the DNA template here, and our clamp head folding over the DNA. So this motif folds into the structured POL4 clamp head, and this is exposed on the enzyme exterior. And this domain is required for POL4 to silence transposons. And the final piece of the question was well, what is the domain important for? Why is it just folding over the DNA that matters? Is that part of the enzyme being able to transcribe and start this whole pathway? It's so highly conserved. We can see that from looking at all these species, from Arabidopsis to mosses. This is the entire range of terrestrial plants. So to better understand that, we did some mass spectrometry at the IBMC in Strasbourg with the platform run by Philipp Hamann. We had mutants for the domain of interest in the wild type. And what we were able to see is that we can find all of the peptides for POL4, all of its subunits here, and the RDR2, that's the RNA-dependent RNA polymerase to the left, and these CLASI proteins I mentioned that are supposed to recruit POL4 in the wild type. We can see all of those peptides easily. But when we have the mutations in our domain of interest here, we're going to lose all of the CLASIs, all of these recruitment factors, so no peptides are detected. So what this means is that this region of the enzyme of POL4 is a docking region for POL4 to interact with its CLASI partners. And if we mutate that region in any way, the small interfering RNAs that we're studying, these are the small RNAs that are produced by the enzyme, are lost in our null mutants for either POL4 or the CLASIs. And we can bring the small RNA levels back up just by bringing in a wild type version with that domain intact, with this docking domain intact. As soon as we make any mutations here, though, we lose all of the small RNAs. So we have a model, a working model for how this works, thanks to AlphaFold and functional genetics that we've done, that shows that POL4 can interact through this domain with CLASI and SHH1. This is a factor that is reading marks in the chromatin, silent repressive marks. And then when this is recruited, we have double-stranded RNA made, small interfering RNAs, and this then causes a targeting of the transposon and a silencing. And another result from the paper that was important was that there are 4 of these CLASIs in our model, in our plant, and we were able to see that none of them could co-occupy this site. So it appears that there are different transcription factors, if you will, for this enzyme POL4, different factors that can recruit the enzyme, and that this is a part of the diversification of its function in silencing transposons. So I think we're over the 45, 50-minute mark, so I'm gonna stop. But we can go into the question session if you want to talk a little bit more about artificial intelligence or the research program that we're running. And I'd really like to thank the people in our lab, especially Louisa, for this wonderful PhD work she's done, and then several other students in the lab that have been working on related projects. The bioinformatics work was done by Calvin, and our lab manager is Christophe Imber. And we have another teacher who you probably recognize in the lab, Fabrice Michel, who teaches courses in licence and in your master degree program. So thank you very much for your attention.
Yeah, so thank you very much. Very interesting, very inspiring. So is there first, as we do it always, is there an icebreaker? Is there some question? I hope so. So I wonder, you said that in the beginning, you guys, you're kind of very active in maybe climate change and all this. Did you see what's happening if you make one ChatGPT? Incredible. So much energy used.
Yeah, I mean, that's, I don't know if this is on, is it?
Yes,
Okay.
it's on, yes.
So the issue, of course, with these models is that they can be so revealing for now people are using them to plan their schedules or plan their study plan or search the web, search knowledge bases. But every time you make one of those requests, obviously you're not only using energy historically for the training of the model, but you're asking the company to do inference for you. So those GPUs, they start lighting up again. Every one of the GPUs from NVIDIA uses hundreds of watts to do these calculations. Of course, you're using a very small amount of that, but it's just something to keep in mind that we used to have a bookshelf full of reference material, or we had a computer hard drive with reference materials. That is a fraction of the cost of making these requests to the large language model.
Okay, so it's just maybe if you're looking for something, it's better to make a Google, just look for Google and not just ChatGPT for these questions, you know, of course not for science, but for normal requests.
The thing is that Google is now integrating AI into their search pipeline, so I guess we don't escape it,
Yeah. Is there some other question? Where? Oh yeah, okay. Thank you.
Hello, so thank you very much for the lecture. So I had two questions.
Sure.
The first one will be about the AlphaFold.
Yes?
I wanted to know, because you said that AlphaFold 2 accurately predicts the structure from sequences.
Yes.
And I would like to know what's the rebuttal, what's the limit of that technology, basically, because I doubt that it's not possible to give it everything and anything. And you showed us that it went from 93 million from 200 million in the next generation. And so with 200 million, what's the limit for AlphaFold 3?
Okay, so those are two questions. I'll take the first one. So you're absolutely right. There are many cases where we'll try AlphaFold 2. So let's take the first version with 93 million parameters. We'll try to run it. It's more of a research tool. We have to run it on servers. It's not something you can just run using a website, unfortunately, and not get the result that we're interested in. In other words, an accurate prediction of our protein. So sometimes it's a particular domain that is intrinsically disordered. So that domain will have what one in slang might say a kind of floppiness. It's not forming a globular structure. It doesn't have maybe only one state that it can be in. It can be in multiple different states. And AlphaFold doesn't do a very good job of predicting those domains, specifically because there isn't just one structure that can be assumed. So long disordered regions are not predicted by AlphaFold well. Another class of protein that's difficult in general to predict with AlphaFold are proteins that aren't in typical model organisms or in the medical context human genome. So they are poorly represented in the genomic databases, in particular poorly represented in the structural databases. And we tend to see that when we try to run AlphaFold on those proteins from ecological contexts or organisms that we don't work with in the laboratory as frequently, we don't get good results at all. So the predictions are very low quality. Trying to think of some other cases. I mean, it's certainly got error modes that you would see. And if you don't have some strong comparative structures from cryo-EM or crystallography, sometimes it's hard to know whether you should believe something. In our case, we know that the RNA polymerases that are sort of the core part of this enzyme POL4 have been resolved by cryo-EM approaches for years now, since the first one was resolved in 2000. So we have about 2 decades worth of structures we can compare our prediction to. And then the next thing you have to do is more experiments, right? So in the laboratory, you might have a structure that you think is interesting, predicted by AlphaFold, but you have to do site-directed mutagenesis, like Louisa Felgin did, to look at which amino acids could be critical based on the structural prediction. The second question was about AlphaFold 3. So the main advantage of AlphaFold 3, I didn't point this out in detail, is that it can do much more than look at a monomer or say 2 or 3 subunit structure, which AlphaFold 2 can look at maybe 2 to 3 subunit structures if you run it on a big server. So AlphaFold 3 is a web service. I mean, we don't have the resources typically to run such big models as academics. You'd have to be really specialized in the field to do AlphaFold 3. And you can see here in this slide, this is one of the web servers that you can use directly from Google. So you input your proteins 1, 2, 3, 4, 5, 6. So I put the entire complex of RNA polymerase 4, which is 12 subunits, and the entire complex of RNA polymerase 2, which is 12 subunits, into the server. And for good measure, I put in DNA. So the first strand and the second strand of a kind of a template typically used in the structural biology context. And an RNA that's not shown here as well. So an RNA molecule that is being synthesized in the original empirical data. And this would be impossible to AlphaFold 2. It doesn't have the size of model that can handle all of this at one time in memory. And this is run in about 10 minutes, just by making a Google search, essentially. So any one of you that's interested in a protein complex interacting with DNA or RNA or other molecules, small molecules, you can do this experiment yourself at this server, this AlphaFold 3 server. And that wasn't possible even a year and a half or 2 years ago. So that's great. I'll have to say, for our particular case, we see the prediction isn't quite as good as AlphaFold 2. So it's possible to make these higher order complexes and make predictions that are of interest. But in the details, AlphaFold 2 sometimes produces a better result, which is interesting. So just to give you an example of what those results would look like, with AlphaFold 3, you can see POL2 on the left with the DNA template and the RNA exiting here. And then POL4 here with the DNA template and the RNA trying to exit but not getting out there. So we can look at the details of these structures, compare them, and again we have metrics to understand the complexes, these higher order complexes of proteins.
Okay, thank you. It shows also that, of course, if you're making drug design, that's the point, that's the thing you need. And maybe one question, if you allow this, I know you are hungry. What is with structural flexibility in this context? Because enzymes are working, they have, you know, how does this influence structure, data, prediction? Or is this just, you know, one moment and then you have to make for the other moment?
Yeah, my impression is you're missing some of that information on the whole, because it can probably provide you one version of that flexibility you're mentioning. If it is a globular protein, if the domains have enough support in the model to be predicted and it's not disordered. But the thing about AlphaFold 2 and 3, both models, that is interesting is you can run them again and again with a different random seed. So you can input into the model a different seed. It's a number that is generated normally for you. And you can run iterations. So there's something that was developed in France by a group. It's available on the Institut Français de Bioinformatique, the IFB where you can now run AlphaFold 2 and 3, by the way. And in that server, that national infrastructure, you can choose to run the model 100 times. 100 different times, and then you can compare, and maybe I think maybe you would incorporate potentially different versions of the enzyme or the multi protein complex. Of course, this is purely predictive, and you would have to do some work in the laboratory to see if it's credible.
Okay, thank you very much once more. Maybe show me your last slide. This just wants to show you that I always say go to international, makes internships. You also have this research here in Strasbourg at the IBMC, at the IBMP, the IGBMC, and also IPCB. So don't hesitate to contact those people. They are happy to welcome you if you want to make some hands-on learning because you didn't do enough during your studies. So don't forget, they are there, open.
Yeah,
they have open heart.
That's true if someone is interested in, especially a very ambitious L3 student, to do a summer internship. So it's a bit late to be getting the convention de stage at this moment.
Yes.
Or in the master program you might be entering.
Yeah. And
Please
I see there are some of the students we had some years ago. So
yeah, we've had some very excellent master students from your faculty here.
Yeah. Okay, so thank you very much once more, Todd. I wish you a nice evening, bon appétit, and if you have questions, just come on stage as always and you chat with Todd. Thank you very much.
Thank you for your attention tonight. Bye.
And in 2 weeks we have our control continue. You will be informed in time. No problem.